Welcome to Module 13. This is Kim Koska, and I'm going to give you a brief overview of nucleic acids and carbohydrates to get you started. Let's first take a look at the Module 13 overview. So as usual, there are some introduction and some learning objectives to read, and then you'll notice there's activities and 2 assignments. Just a note about the activities are that you'll notice these are the same readings that were assigned in Module 12. So you may have already read these, and so you're ahead of the game if you did. Chapter 18 and Chapter 17 were focused more on proteins and lipids. And in this module, we're going to focus more on nucleic acids and carbohydrates. So our focus is going to be mainly on Chapter 16 and Chapter 19. That said, you will need all 4 of those chapters to perform on the Module 13 quiz. So let's go ahead and take a look at the work you'll be doing with nucleic acids and carbohydrates as you prepare to create a slide deck on nucleic acids. So first of all, we'll just dive in and look at the nucleic acid DNA. So if you look at DNA, we're going to start looking on the right with the chromosome. So that blue X is a chromosome, a representation of a chromosome. And if we uncurl the chromosome, which is made of tightly coiled DNA, we see the structure unfold. So first of all, you might notice that the structure is twisted, and that's called a helix. You'll also note it looks like there's rungs of a ladder between 2 sides of the helix, so it's called a double helix. And those 2 sides are connected to each other by what are called nucleobases. And if we look at the structure there in the middle with the colors, you'll see that there's this nucleobase labeled C, G, A, and T, and those stand for cytosine, guanine, adenine, and thymine. They're connected to each other through space by hydrogen bonds, which is that intermolecular force that you've already studied. So the little dotted lines between them are representing that hydrogen bond that's knitting that whole double helix together. You'll also notice that the backbone is made up of these S and P functions. So that P group little p stands for phosphate, and the S stands for a sugar, which in this case is deoxyribose. So let's take a look now at DNA nucleic acid versus RNA nucleic acid. So DNA, that double-stranded helix, is represented on the left, and we can see some facts about ribonucleic acid, RNA, on the right. So DNA abbreviation comes from deoxyribo, N from nucleic, and A from acid. RNA is R from ribo, N from nucleic, and A from acid. So their names tell us a little bit of something that's a key difference, and that is that they have 2 different kinds of sugars that are related to each other. RNA is based on the sugar ribose in its backbone, and DNA on deoxyribose in the background. So how those are different is that there's 1 oxygen missing for the deoxyribose. Kind of makes sense, right? Deoxy, we've lost an oxygen, and ribose has that extra oxygen. Another key difference, so the different base pairs that we talked about earlier are present in DNA and RNA. You'll see that the 3 that they have in common are listed below: cytosine, guanine, and adenine. But on the RNA side, you'll see that the other nucleobase is uracil, whereas in DNA on the left, it's thymine. And those actually have very similar structures. They only differ by the CH3 group in that position to the left. So you can see that there's a difference there. So Mother Nature is very clever about being efficient. These molecules, deoxyribose and ribose, differ only by 1 oxygen, and thymine and uracil differ only by 1 carbon, and yet their functions are hugely different. So let's take a look again at our structures. So in our DNA molecule, we had those um, phosphate sugar backbones and then the nucleobase pairs coming off of it. So let's blow one of those up. So we're going to look at the adenine example. So we can see phosphate. So again, PO4, you're familiar with that from our module 3 days. And then we've got that chemically bonded to a sugar, in this case deoxyribose, and that itself is chemically bonded to this nitrogenous base, adenine. So when we look at the bases, you're going to see some familiar functional groups. So you see amines, right? NH2 groups here and there, and NH groups as well, even those that are embedded in the ring. You can also see amide groups, right? So we've got carbon double bonded to oxygen as a neighbor to a nitrogen atom. So amines and amides are the basic functional groups that we see in these 4 molecules. So these are the molecules of DNA. Let's look at the molecules of nucleobases of RNA. So instead of thymine, we're going to replace that again with uracil, and we talked a little bit about that already. So let's go ahead and put those back together. The reason I'm showing you lots and lots of visuals here is because when you get to Module 13 quiz, you'll be seeing lots of structures and be asked to identify them. Or you might be given a name and then select a structure that matches that name. So we've got, again, our phosphates attached to sugars, attached to the nucleobases. So here's, since we have thymine on this screen, we've got for sure the bases of DNA, and also because we've got deoxyribose, these are DNA structures. So let's take a look at the sugars in more detail. So this sugar, ribose, has 5 carbons, so it's called a pentose, pent for 5, and then ose is the name that the suffix that we use for sugars. So anytime you see the name of something ending in ose, we're talking about our carbohydrate. But there are other kinds of oses or carbohydrates. So here are structures for glucose and mannose, which are 2 common carbohydrates. Glucose and mannose are both hexoses. They have 6 carbons in each of them. So 6 is hex and ose is carbohydrate. So 6 carbon carbohydrates. There are a couple of common ways that we display the structure of a carbohydrate. One is to show it in the ring form, which you've already seen. And here are the ring forms for glucose and mannose. By the way, the ring form can vary a little bit at carbon 1 position if the OH group is up or down. So we have different names for that, alpha and beta. You don't need to know that kind of detail when you're working with your quiz this week, but just to explain what you're seeing on the slide. You can also see at the top of the slide that we can also draw the sugars as a straight line. So that's yet another common way that we'll see carbohydrates represented. So far, all of the carbohydrates we've looked at, all of these sugars, we've looked at are monosaccharides. Mono meaning 1 and saccharide meaning sugar. So 1 sugars. We can also find other kinds of sugars in our everyday life. So on this slide, you can see glucose, fructose, and galactose on the top left. Those are examples of monosaccharides. Ribose is another, and deoxyribose yet another, all monosaccharides. Then on the left or the right, we've got sucrose, which is a disaccharide of glucose and fructose chemically bonded to each other. And at the bottom, lactose, which is galactose and glucose chemically bonded to each other. So those are disaccharides. We can also have polysaccharides. So the structures that you see on the lower left, cellulose, starch, and glycogen, are polysaccharides. So we've got cellulose and starch are both produced by plants, and cellulose we would think of as fiber, or the thing that we would eat is fiber, or even wood in trees as fiber. Glycogen is a polysaccharide that we find in our bodies. It's stored in the liver and is used for energy, and it's a polysaccharide of glucose. So the sugars do vary over all of Mother Nature, and we can see quite a lot of variety in the kinds of carbohydrates that are available to us in foods and in our environment. So going back again to our assignments for this week, there's an assignment overview video that will be helpful to you, but just a reminder that for the Module 13 nucleic acid slides, you'll be focusing on Chapter 16 and 19 from your textbook, and for the Module 13 quiz, you'll be focusing on identifying structures, and those will include structures both from Module 12 and from Module 13. So best wishes to you as you work through this module, and please remember that we are always available for questions. If you have them, send them along in our inbox or post them in discussion. You can also certainly attend office hours, and we would love to see you there.